Hello, very good morning to all of you. Dear friends, we have started the protein characterization part of this food analysis section. In last lecture, we have started the protein functional properties, and in that we know that there are mainly three protein functional properties are there. First one is solubility, second one is emulsification, third one is foaming. Okay, and two subsidiary protein functional properties are there, and these are gel formation and dough formation. The first two are over. Now let us see the third protein functional property, and that is foaming. So just like emulsion, foaming is also important for the food products. Okay. So what is the principle of this foaming process? What is foam? Foam is a coarse dispersions of gas bubbles. Coarse dispersions of gas bubbles. Coarse means which is not fine. Okay, which is not fine. It is called coarse. A coarse dispersions of gas bubbles in a liquid or a semi-solid continuous phase is called a foam. What is foam? A coarse dispersion of a gas bubbles. So gas bubbles are incorporated in a liquid. It is called a foam, or it is a semi-solid continuous phase. This is known as foam. Now, the methods for foaming process are generally three main important processes are there. These are whipping process, shaking process, and sparging process. So what is whipping process? Whipping process means whip, that is a rope like material, and that is used to beaten. It is known as whipping process. Okay, so a stick or a rope is generally used to beaten that material, that solution. Okay, and this process is called as whipping process. Shaking, you know that shaking process. And sparging. Sparging means introduction of gas, injection. So gas is injected into the liquid. That process is called as sparging process. So these are whipping, shaking, and sparging are the three main important processes for the foaming process. Now why we are discussing here under the protein heading? Because the proteins are important part in this foaming process of food products. Because the proteins lowers the surface tension. Proteins lowers the surface tension between the two phases, that is a liquid phase and a gas phase. Because gas bubbles are incorporated into the liquid, hence it is again a type of colloidal material. Foam is one of the type of colloidal material, and in this continuous phase is a liquid phase and dispersed phase or discontinuous phase is a gas bubbles. Okay, and again there is a boundary is formed between the two phases. One is gas phase and another is a liquid phase, and these protein molecules are lowering the surface tension at the boundary of gas bubbles. Okay, and we know that as there is a boundary between gas bubbles and the liquid phase, so these protein molecules are helpful for lowering the surface tension of these gas bubbles with the continuous liquid phase. Okay, and hence during foam formation this process happening of lowering the surface tension, and hence these protein molecules are useful or they are helpful for imparting the stability in the foam formation or imparting stability to the gas bubbles in the foam formation process. Okay. So this is the principle of foaming. Then procedure. So two important parameters are used to evaluate the foaming ability of a protein. The foaming ability of protein can be evaluated by the two important parameters, and these are foam volume and foam stability. Foam volume and foam stability. Now what is this foam volume? Volume of a foam. So during process of foaming, this volume of foam is determined. Okay, and this foam volume depends on the ability of foam to lower the surface tension. So foam volume is responsible for lowering the surface tension of that foam by that protein. Okay, the foam is formed in a blender. The foam is generally formed in a blender, and it is transferred to a measuring cylinder. From which we can measure the volume of a foam. Okay, so measuring cylinder has a marking, and from that we can measure the volume of foam in milliliter or liters. Okay, so foam is formed during foaming process. Foam is formed in a blender, and then it is transferred to a measuring cylinder, from which we can easily measure the volume of that foam. Then second one parameter is foam stability. So foam stability is another important parameter for the foaming process of a protein. The foam is placed in a funnel. Oh, a funnel is there. Okay, so the foam is placed in a funnel, and below that funnel a measuring cylinder or graduated cylinder is used. A graduated cylinder is placed. Okay, and over this graduated cylinder this foam is placed, and in that foam funnel the foam is placed. Okay, then no doubt the foam that we have placed over this funnel, it will start to drain from this funnel into this graduated cylinder or measuring cylinder. Now we have to count or measure the time to drain the foam from this funnel in this graduated cylinder, and half the time required for draining this foam, from this, for draining this foam from this funnel to this graduated cylinder is recorded. Okay, and the greater the half time, more stable is the foam. Greater the half time, more stable is the foam. Okay, so this is related to the half time method for the foam stability, and foam stability is evaluated by using this funnel and graduated cylinder method. It is very simple. Just once the foam is prepared, it is formed, it is placed over this funnel, and this funnel is taken over this graduated cylinder. Okay, then measure the time required for draining this foam from this funnel to this graduated cylinder, and half time required for half the volume of that foam is recorded, and it is called as T1 half. Okay, and higher the half time, greater the half time of that foam drain process, more stable is the foam. So this is the relation between foam stability and the time of flow for that foam in the graduated cylinder. So here the three important protein functional properties are over. Then the two subsidiary properties are gelation and dough formation that we have to see here. So what is gelation? Gelation is nothing but gel formation. So what is gel formation? Protein gels are formed, or they are made by treating a protein solution with heat, enzymes or divalent cations. So how protein gels are formed? Gel is another type of colloidal material. Okay, just like emulsion, foam and gel, these are the different types of colloidal material. Okay, and in all these types of colloidal material there are two phases always. One is continuous phase, which is in larger volume, and another is discontinuous phase, which is in lower volume, or one is called as dispersed phase, which is in lower volume, and other is called as continuous phase. Okay, and in this gel, protein gel are formed by treating a protein solution with either heat, enzymes or divalent cations. Some protein gels are formed by giving a heat treatment to them. Some protein gels are formed by enzymatic reaction known as proteolysis, and some protein gels are formed by treating these protein solutions with divalent cations such as calcium 2 plus or magnesium 2 plus, Then during gel formation or gelation process, proteins are transferred from soluble state to cross-linked gel-like state. Already in protein solution, the proteins are in soluble state, but when gel formation or gelation process has been occurred, then there is a change of state from a soluble state of protein to the cross-linked gel-like state. Cross-linking is there. Okay, and in that cross-linked gel-like state of that protein, it may involve covalent or non-covalent interactions. So covalent interactions are generally disulfide bonds. These are covalent interactions in the cross-linked gel-like state of the protein. And non-covalent interactions including hydrogen bonding, electrostatic forces of attraction, then hydrophilic nature, hydrophilicity or hydrophobicity, all these considered under the non-covalent interactions. Hydrophilicity, hydrophobicity, then an electrostatic attraction and the hydrogen bonding. Okay, they are considered under the non-covalent interactions. Some protein gels are thermally reversible. Means what? When we are giving heat, then they can be again converted into soluble form. So cross-linked protein are again converted to soluble protein in protein solution. Okay, so that's why they are called as thermally reversible. But some of the protein gels are thermally irreversible. Means if we are giving heat treatment to them, then also there will be no any change from the cross-linked protein to the soluble protein. The stability of protein gel depends on nature and concentration of protein. So this is one of the important factor. Then temperature of the gelation process, then pH of the medium, then ionic strength of that solution. Okay, all these factors are responsible for this stability of protein gel. Then the next and last point from this chapter and from this protein functional properties is dough formation. Dough In Marathi we call it as a kanik. In Marathi we call it as a kanik. So what is dough? When a flour is mixed with water, for example we have a wheat flour, wheat, gehu ka pit, okay, so we have a wheat flour, and when a water is added to that wheat flour, then what is the result that we all of you are known to us? That is that wheat flour is get converted to a dough. We call it as a dough. And once dough is formed, with the help of hands, our fingers, we need that dough and convert it into a dough. Okay, with the help of kneading process we convert it into a dough, and that is helpful for making different bakery products as well as for making rotis. Okay, so here the wheat protein is unique. Wheat protein is unique to form viscoelastic dough. Viscoelastic means it is a property of that protein or that dough material. It is called as visco. It has some viscosity and it is elastic in nature, means when we stretch that material, when we add water to that wheat flour, then after kneading process, that resulting form of that material is called as dough, and it has viscoelastic properties. When we stretch that material, it elongates, so that's why it is called as a viscoelastic dough. Okay, and it is useful for the making of bread and other bakery products. Okay, so this wheat protein is mostly important part in this dough formation. Okay, so gluten is the responsible, or which is an important protein present in the wheat flour. And what is this gluten? Gluten is a heterogeneous mixture. So it is not a homogeneous, it is a heterogeneous mixture of two proteins known as gliadins and glutamines. So gluten is a heterogeneous coarse mixture of gliadins and glutamines. So these two proteins are present in heterogeneous manner in that protein, and that protein is known as gluten, and this gluten is one of the major storage protein in the wheat flour. Gluten is one of the important and main storage protein in the wheat flour, which is a combination of or heterogeneous mixture of gliadins and glutamines. It makes possible formation of viscoelastic dough, which can trap the CO2 gas, carbon dioxide gas, during yeast fermentation. So during yeast fermentation means this is related to the food industry or food processing. It is not actually there is nothing chemistry in this, but this is related to the food processing activities. Okay, so during baking activities, we know that yeast is used for the fermentation process for making different bakery products, and in that bakery processes or activities, that carbon dioxide gas is trapped between this gluten protein. Okay, and this is responsible for the formation of viscoelastic dough. Okay, the dough strength, once this dough is formed, this dough strength is measured by either mixograph or farinograph. So dough strength is measured by either by mixograph or farinograph. So mixograph is generally used to test mixing properties of flour. So when we mix the flour, okay, so that mixing properties of flour is tested by a mixograph. The name itself indicate it is a mixograph. And the another is nothing but farinograph, and farinograph is used to test water absorption ability of the flour. when we add water to that wheat flour, so water absorption ability of that flour is get tested by that farinagraph, and both these mixograph and farinagraph are used to determine the dough strength, and this dough strength is important parameter for the dough formation in the bakery products. Okay, so that's why in this way we have studied all these 5 properties. The two already completed. This is third one, foaming, fourth one is gelation, and fifth one is dough formation. Okay, these are the protein functional properties. Okay, and in this way we have also completed our 6th chapter, that is protein. The protein per chapter was divided into 2 sections, 2 parts. First part was related to the protein analysis, and the second part, or the remaining part, half section, was related to the protein characterization. Okay, so in this way we have completed both of these parts. Okay, and in the next lecture we will start the next chapter, that is carbohydrate analysis. Okay, thanks for watching this video. Have a nice day.